Hello everyone and welcome to my YouTube channel Dr. in White. Today we will talk about the secondary messengers and our main focus will be on cyclic AMP and cyclic GMP. So here we begin. First we will learn about the secondary messengers. What are secondary messengers and how they function. So before that we should know about the first messengers. So hormones act as a first messenger of all the biological system. That means hormones हमारी body में as a first messengers act करते हैं. They interact with the membrane receptors and control most of the major body functions, from simple basic need like hunger to even the complex body function like reproduction. ये सब हमारी body में hormones control करते हैं. So peptide hormones are basically hydrophilic molecules. So they are water loving molecules. So they are unable to cross the phospholipid bilayer of the plasma membrane. हमारी जो सेल की लेयर होती है they are basically known as the plasma membrane. They are the phosphobilipid layer. That means they are made up of lipids. And as if we know that hormones that are peptide hormones, they are hydrophilic. So they are unable to cross this membrane. So for that, in this case, these hormones interact with the specific receptor molecules. यानी कि सेल पे एक specific receptors होते हैं जो हार्मोन से जाके अटैच हो जाते हैं and they stimulate the release of intracellular signaling molecules by the cells and they trigger the physiological changes like they activate the cell to do that function particularly, and this intracellular signaling molecule that causes the physiological response that produces a change in some activity of the cells are called secondary messengers. So जब हार्मोन हमारे रिसेप्टर पे आके बाइंड होता है, so it forms a signaling molecule in our cell which will further produces changes in the cell and let it do its function. So this molecule is known as secondary messenger or second messenger. So first messengers हमारे hormones है और second messengers वो है जो basically hormones और receptor का interaction से एक molecule generate. So now we will see secondary messengers type. So secondary messengers, if they are a cyclic nucleotide compound, they can be cyclic AMP, they can be cyclic GMP, and if they are a phospholipid derivative, they can be diacylglycerol or they can be inositol triphosphate. And the third type is another system that operates in response to the entry of the calcium into. So they basically acts when we have a entry of a calcium into the cells. So this is calcium calmodulin second messenger system. So we have three systems: cyclic nucleotides, a derivative, and another system that works on the entry of calcium ion. So now we will see the responses. The receptor provides the recognition for the hormone cell interaction, and this binding event must be translated into the cellular response binding of the hormone to its membrane receptor, and often results in activation of nucleotide cyclizing enzyme located in the inner surface of the membrane. So this means that a cell is always having a receptor for all those hormones which are hydrophilic hormones, and they cannot act inside the cell by direct entry. So they needs a receptors on the cell membrane so these hormones will act on these receptors and form a hormone cell interaction complex, and this binding will be converted into a cellular response that will be a secondary messenger, and this mainly result in the activation of the nucleotide cyclase enzyme located on the inner surface of the membrane. Enzyme adenyl cyclase, upon the stimulation by the chemical messengers, convert ATP to the cyclic AMP, that is 3,5-adenosine monophosphate. So in the membrane we have a adenyl cyclase enzyme located on the inner membrane of the cell, and this is activated by the conversion of ATP to cyclic AMP. So this enzyme adenyl cyclase, when it is stimulated by the action of like hormone cell interaction, it converts the ATP to the cyclic AMP, that is 3,5-adenosine monophosphate. And some hormones activate guanylate cyclase that converts GTP to the cyclic GMP. So we have two responses. First, when the adenyl cyclase enzymes works on the ATP to convert it into cyclic AMP, and we have a second response in which the hormone activates guanylate cyclase that converts GTP to the cyclic GMP. So as we have talked about that every cell needs a special receptor so that the hormone can act upon that and forms a complex. So these receptors are known as G protein linked receptors. So GPCR, that is G protein coupled receptors, that shares a common structural signature of a 7 hydrophobic transmembrane. So they are basically like this. They form a 7 loops on the membrane like this. So they have a special name called 7 hydrophobic transmembrane with an extracellular amino terminase and an intracellular carboxyl terminase. So that means on the extracellular surface they have the amino terminase, and on the inner surface they have the carboxylase terminase. So G protein coupled receptors activate a class of membrane bound protein which then migrates into the plane of the plasma membrane, initiating the cascade effect, and this result in the enzymatic alteration and generate a host of additional signals called second messenger. सो दिस जी प्रोटीन लिंक्ड रिसेप्टर्स, दैट मीन्स ये एक्टिवेट करता है एक तरह के प्रोटीन को विच जो बाद में जाके माइग्रेट होता है प्लेन ऑफ द प्लाज्मा मेम्ब्रेन में, जो फर्दर इफेक्ट जनरेट करता है जिसके थ्रू हमारा सेकेंडरी मैसेंजर्स जनरेट होते हैं. So we will see the scheme of this, like G protein linked receptors bind to the extracellular signal molecules, that is hormones, and this is known as the primary messengers or the first messengers, and this pass to the GTP binding protein associated with the receptors. So by this G protein gets activated and it leaves the receptors, and this turns on the target membrane in the plane of the plasma membrane. And when the G protein is activated, it dissociates into signaling प्रोटीन्स. सो जब ये जी प्रोटीन एक्टिवेट हो जाता है तो ये एक सिग्नलिंग प्रोटीन जनरेट करता है. And after the regulation of the target protein is completed, the G protein alpha subunit switches off by hydrolyzing the bound GTP into GDP by GTPase. And then the reassociation of alpha and beta gamma complex takes place, making the G protein ready to couple with the other receptor. So G protein couple linked receptors, it is a 7 hydrophobic transmembrane segment. So we can clearly see these 7 membranes like 1, 2, 3, 4, 5, 6, 7, and these also have their subunits like alpha, beta and gamma. So here comes our hormone, and it will go inside or react with the receptor. Alpha subunit has a GDP in initial. So when this alpha unit has GDP linked with it, it is a switch off state of this receptor molecule. But when this GTP that binds to the alpha receptor and taking off the GDP, and this alpha receptor goes with the GTP, is known as switch on phase of this receptors. And when the molecule is released, then alpha and beta receptor will allow the alpha unit with the GDP to bind again, and this complex is also known as switch off. So now we have to differentiate between switch off and switch on G protein couple receptors. तो जो हमारी जो फर्स्ट फेज था जिसमें कि GDP अल्फा सब यूनिट से कंबाइंड थी, so this state is known as switch off, जिसमें GDP अल्फा सब यूनिट से bind है. And in fact the second stage may be GDP bind, this is also a switch off. But in the third state GDP will goes and binds to the alpha subunit, and then in the fifth phase that GTP is binded to the alpha state and leaves the membrane as a G protein, and in the fifth state we can say that it is a switch on phase of the whole cycle. Next we will see the requirements for a compound to act as a second messenger. So during a hormone receptor binding, the concentration of the second messenger should increase in order to exert the biological effect. That means जब एक हार्मोन रिसेप्टर से बाइंड हो रहा होता है तो उसकी कॉन्टिन्यूसली कॉन्सेंट्रेशन बढ़ती रहनी चाहिए, जिससे कि हमारा बायोलॉजिकल इफेक्ट भी स्पॉन्टेनियसली इंक्रीज होता रहेगा. Whereas when the hormone is removed, the concentration of the second messengers and the biological response shown by the hormone should decrease in the concentration and in the activity. ऊपर वाले में देखा जैसे जैसे हार्मोन रिसेप्टर की बाइंडिंग बढ़ती जाएगी वैसे वैसे हमारी बायोलॉजिकल इफेक्ट भी बढ़ता जाएगा, बट जैसे हमारी हार्मोन कम होता जाएगा वैसे वैसे हमारा बायोलॉजिकल रिस्पॉन्स भी कम होता जाएगा. So now we will see in detailing about the cyclic AMP. So now we will see the steps involved in the cyclic AMP. First, the binding of a ligand to a specific receptors in the cell membrane. अभी हमने देखा कि एक हार्मोन को एक स्पेशल रिसेप्टर चाहिए क्योंकि वो एंटर नहीं कर सकता सेल में. सेकेंड, द एक्टिवेशन ऑफ अ जी प्रोटीन आफ्टर द फॉर्मेशन ऑफ अ कॉम्प्लेक्स जीडीपी इज रिप्लेस्ड बाय अ जीटीपी. लास्ट स्लाइड में देखा कि जी प्रोटीन का एक्टिवेशन हुआ जब जीटीपी ने जीडीपी को रिप्लेस किया. इन द थर्ड स्टेप देयर इज एक्टिवेशन ऑफ द एंजाइम adenylate cyclase that activated G protein either stimulates or inhibits the enzyme adenylate cyclase which is located in the plasma membrane. So जो G protein हमने अभी रिलीज हुआ वो या तो एंजाइम एडिनाइलेट साइक्लेज को इनहिबिट करेगा या फिर स्टिमुलेट करेगा जो कि सेल मेम्ब्रेन के अंदर लोकेटेड है. Step number 4, formation of the cyclic AMP. When adenylate cyclase activated, it catalyzed the formation of cyclic AMP from cytoplasmic ATP with magnesium ion as a cofactor. That means cytoplasmic ATP will convert into the cyclic AMP with the magnesium ion as a cofactor. Thus, a stimulatory G protein will increase the level of cAMP, whereas an inhibitory G protein will decrease the level of the cAMP. ऊपर हमने देखा कि जी प्रोटीन या तो जी प्रोटीन स्टिमुलेट करेगा एडिनाइलेट साइक्लेज एंजाइम को या फिर ये इनहिबिट करेगा. अब अगर जी प्रोटीन इनहिबिटरी नेचर का है तो वो CAMP का लेवल डिक्रीज कर देगा. If it is a stimulatory G protein, then it will increase the level of the CAMP. Step number 5, action of the cyclic AMP. It activates the protein kinase A, that is pKa. Then it phosphorylates the other leads to the cell response. So now we will see this in more detailing. So this is our cell membrane. So here we have a cell receptor. So the ligand will bind to the cell receptor and will release a G protein which can be stimulatory or it can be inhibitory. So this will affect the level of the adenyl cyclase. So cytoplasmic ATP which is located inside the cell and cytoplasm. This cytoplasmic ATP in the presence of adenylyl cyclase enzyme will convert into cyclic AMP, and this will further be inactive or active depending upon the presence of protein kinase A. So if it is having a protein kinase A, this can be in active state resulting in a cellular response, but if protein kinase A is not active, it will result in inactivation. So simply we can state that ATP in presence of adenyl cyclase will give rise to cyclic AMP. So now we will see the termination of cyclic AMP. The action of cyclic AMP in the cell are terminated in two ways. Cyclic AMP is degraded to 5-AMP in the cell by the cytoplasmic enzyme phosphodiesterase. So that means if in our cytoplasm we have the enzyme phosphodiesterase, then the cyclic AMP will be converted into 5-AMP. And specific phosphates dephosphorylates the protein already phosphorylated by the protein kinase A. Now we look at the functions of cyclic AMP. It altered the glycogen metabolism by the enzyme regulation. So it controls or alters the glycogen metabolism. It will alter the gene transcription. It is responsible for the hormone secretion like estradiol. It altered the ion transport like HCl secretion in the stomach. It is responsible for the water reabsorption by the antidiuretic hormones, that is ADH hormone. Now we will see that some hormones that uses adenylase cyclase, that is cAMP, as a secondary messengers, that can be adrenocorticotropic hormone ACTH, angiotensin II, mainly the epithelial cells, the calcitonin, the catecholamines, that is beta receptors, the corticotropin releasing hormone, the follicle stimulated hormone FSH, glucagon, human chorionic gonadotropin HCH, HCG, luteinizing hormone LH, parathyroid hormone, secretin, somatostatin, thyroid stimulating hormone, vasopressin, mainly the epithelial cells. Now we will talk about the cyclic GMP. So the signaling pathway that regulates the synthesis of cyclic GMP in cells include the hormonal regulation of transmembrane guanylate cyclases such as atrial natriuretic peptide receptors, that is ANP, and the activation of the soluble forms of guanylate cyclase by the nitric oxide. So as if we seen in CAMP that enzyme adenylate cyclase will regulate the all mechanism. So in cGMP the enzyme guanylate cyclase will do all the things. In cyclic GMP the enzyme guanylate cyclase will do all the things such as atrial natriuretic peptide receptors, the activation of the soluble forms of the guanylate cyclase by nitric oxide. Unlike cAMP, GMP has established the signaling roles in only few cell types. So signaling pathways that regulate the synthesis includes nitric oxide and the hormonal regulations like ANP, BNP and CNP. So we will talk about the nitric oxide. So nitric oxide is a gas which diffuses through the plasma membrane and affects the nearby cells. Synthesized from arginine and oxygen by the NO synthesized from arginine and oxygen by the nitric oxide synthase. So here we see that it is a cell membrane. So nitric oxide is a gas that can diffuse from the cell membrane and enters the cell. So with the help of oxygen and arginine, it will form a nitric oxide synthesis. The nitric oxide then activates the soluble guanylyl cyclase to produce cGMP. So this nitric oxide synthesis will produce cyclic GMP with the help of GDP. So the steps involved in this, like function of nitric oxide in the dilation of the blood vessel. So it has a basic function of dilation of the blood vessels. The acetylcholine, that is a neurotransmitter, acts on the endothelium cells to stimulate the nitric oxide synthesis. Nitric oxide then diffuses to the neighboring smooth muscle cells where it interacts with the guanylyl cyclase. Like you can see that this is an endothelium cells and this is a smooth muscle cell. In endothelium cell, with the help of arginine, the nitric oxide will enter into the smooth muscle cells where it interacts with the guanylyl cyclase, which with the help of the GTP will produce cGMP, and this further will result in the relaxation of the smooth muscle cell, and this increase the enzymatic activity result in the synthesis of cGMP. The cGMP then induces the muscle relaxation and blood vessel dilation. Now we will see the types of isoforms. Isoforms are basically the types of nitric oxides present in the different cell types. So first we will see in neuronal type 1 isoform, that is NNOS. So they are present in the CNS nerves, the skeleton muscles, and they perform a role in cell communication as it is a neuronal and is associated with the plasma membrane. Second is inducible type 2 isoform, mainly present in the macrophages, Kupffer cells, neutrophils, fibroblasts, vascular smooth muscle cells, and endothelium cells, and involved in immune defense against the pathogens. Third type is endothelial type 3, basically located in the endothelium, cardiac myocytes, renal mesangial cells, osteoblasts, platelets, and is involved in regulating the vascular function, that is vasodilation. Fourth type is mitochondrial isoform. In this, the nitric oxide in the mitochondrial matrix may regulate the ATP synthesis. So now we will see the role of cyclic GMP in first the visual cycles, second the vasodilatory effect by the nitric oxide, third the activation of the peptide hormones like ANP, BNP, where the membrane bound guanylate cyclase is used. Now we will see the difference between the cyclic AMP and the cyclic GMP. So first difference is cyclic AMP is a common second messenger involved in signaling transduction cascades, whereas cGMP is less common. Second difference is its in name, that is 3,5-cyclic adenosine monophosphate, whereas cGMP is 3,5-cyclic guanosine monophosphate. Third difference is cyclic AMP. It is a adenosine nucleotide, whereas cGMP it is a guanosine nucleotide. It's synthesized from the ATP by the action of adenylate cyclase, whereas cGMP it is activated from GTP by the action of guanylate cyclase. Fifth difference, it has a high concentration in many tissues, whereas cyclic GMP has lower concentration in many tissues. Number 6, cGMP takes part in the regulation of glucagon, sugars, lipids, metabolism in eukaryotes, whereas cGMP takes part in the regulator of ion channels conductance in glycogenolysis, cellular apoptosis, smooth muscle cell relaxation, and phototransduction. Thank you guys for watching my video, and if you like this video, do subscribe, like and comment. The doctor invite. Thank you.